Thank you, Dr. Nee, for the introduction. So my project is the change in androgen receptor activity due to PXR activation via rifampicin. So here's a little background so you can understand what I'm talking about a little bit. So Dr. Nee's lab focuses on prostate cancer, and so the main definitions I want to get down are first androgen receptor. This is a nuclear receptor, and it is a ligand-dependent transcription factor that regulates the genes that are involved in development. So the two cells I worked with here were LNCAP FGC and 22RV1 cells. So the LNCAP FGC cell line, this consists of the full-length androgen receptor, and the 22RV1 cell line has both the full-length androgen receptor and the shortened variant V7AR. So this variant does not contain the binding region that the full-length androgen receptor contains. So the portion it's missing would be the binding region. So this would be what like castration-resistant cancers consist of, because they don't respond to the therapies that target the binding site. So the treatment I was working with was rifampicin, and it's an antibiotic. And the main outline of this would be rifampicin would activate PXR, which would then go on to decrease the activity of the androgen receptor, which would hopefully then decrease the downstream gene expression of the androgen receptor. So I first started with QPCR, and this is to measure the expression of the genes that are regulated by the androgen receptors. So this is the process of what I did to get my results. And so first I would seed the cells into the well plates. I would treat the cells with rifampicin. I did 0, 1, 5, and 25 micromolar concentrations. And then the next day I would extract the RNA from the cells, measure that concentration, use that concentration to calculate equal amounts of nucleic acids, and then I would reverse transcribe that RNA to cDNA, and then add primers to that cDNA and put that into the QPCR machine, which would then, the machine basically denatures the DNA, the primers attach, extend, and that cycle repeats until you get it's called a CT value. And so you can compare that to the control and calculate the full change in gene expression. So this was my first attempt with the LNCAP FGC cells, which is, again, the full-length androgen receptor. So the downstream expression of genes was decreased overall. As you can see, this would be the control, and then increasing concentrations in each one. There was a trend was lacking, so I redid this experiment so I could try to get better results. And so with my second attempt of the same thing, this was similar to the first, but the downstream gene expression was also decreased, and there was a better trend with more significant p-values. And so that was for LNCAP. And then I moved on to 22RV1, which again has the full-length androgen receptor as well as the shortened variant. So these are the same genes to measure the full length as the LNCAP, and it also had similar results where the gene expression was decreased. So when I looked at the V7AR, which would be the shortened androgen receptor without the binding region, there were similar results. So they showed some decrease in gene expression, but as you can see here, this one was kind of an outlier and didn't decrease with it. And this could be due to many other factors as to why the downstream gene expression wasn't decreased, but the fact that there were signs of a decrease in gene expression without the binding site is significant, because this shows that rifampicin could potentially be repurposed to decrease the gene expression of androgen receptors that don't have the binding site, so ones that are resistant to treatment that is commonly used today. So after QPCR, I then went on to do Western blot, and this is the background of Western blot to get an idea of what I did. I first seed the cells into well plates again. I treat them. I did 0, 5, and 25 micromolar concentrations of rifampicin, and then I create the gel, and then the next day I would insert the proteins into the gel, do gel electrophoresis, and then I transfer that to a membrane, attach antibodies with fluorescence that could then be used to analyze the proteins. So this was my first results. This was just 22RV1. And as you can see here, it's very faint, but this would be the androgen receptor V7, so the shortened one with increasing concentrations. And you can kind of see this next slide you can see it more clearly. So this is LNCAP FGC with the full length and 22RV1 with both full length and shortened. So across the top here at about 110 kilodaltons, you can see the full length, and with increasing concentrations, you can see that the proteins seem to remain consistent as concentrations increase. So this is significant because it tells us that the actual protein, the androgen receptor, isn't being destroyed, and the actual activity of it is what's causing the gene expression to decrease. It's not the actual protein being destroyed, which would cause the gene expression to be decreased. So then from here, I did colony formation assay where this is the background of it. Basically, I count the prepared cells, I dilute the sample, and transfer equal amounts into the well plates. This we did 200 LNCAP FGC cells per well. And then we treat the cells. These are my concentrations used. And then I used crystal violet to stain the colonies that formed, count them, and then analyze the data. So as you can see by this graph, the concentration, as the concentration increased of the treatment, the number of colonies decreased. So at smaller concentrations of rifampicin, colony size decreased. And with large concentrations of rifampicin, colony formation was limited and even killed the cells off. So combining all three of these, in conclusion, the gene expression resulting from androgen receptor activity was decreased in the full-length androgen receptors in both cell types after treatment, and then the shortened androgen receptor variant in the 22RV1 cells also showed some decrease in gene expression with treatment. Along with this, protein quantity remained consistent with increasing concentrations of treatment, and colony formation decreased with increasing treatment concentrations. So to sum this all up and bring everything together, the significance is the treatment with rifampicin. This is shown to work with both the full length and shortened, and work with the shortened one as well in the V7 androgen receptors. So because the V7 androgen receptor lacks this binding region, the activation of PXR with rifampicin would have to suppress the androgen receptor through something other than its binding site. So with this, in the future, with more research and evidence, rifampicin could be repurposed to treat the androgen receptors that are already resistant to present-day treatments that target the binding site, which if you could manage to get around, I guess, these resistant cells, have something pretty special there. So I'd like to end my presentation by saying thank you to Simmons Cancer Institute, thank you to SIU, thank you to Dr. Nee, and thank you to a student, Shanwei, who really helped me throughout the summer and gave me a lot of tips. And thank you to all the donors. Without your contributions, I wouldn't be here today. And this project was pretty personal to me because my dad actually has prostate cancer. So without donations like your guys' research in medicine, we can have people we love around today. So thank you for listening.
We have questions?
Great presentation, by the way.
Thank you.
Has been said for senior prostate cancer survivors. So,
Amazing.
I want to know, why did you select rifampicin? Why that?
So Dr. Nee's lab, I believe, was working with this before.
Yeah. PXR is called a tryptophan X receptor. Rifampicin is an antibiotic used for treating for TB. And is a take over this stuff. But rifampicin is the activator of TXR. So since this rifampicin is commonly available, it is non-toxicity profile, so we wanted to see whether we can repurpose this drug for prostate cancer treatment.
Go ahead.
What about rifampicin allows it to get around those binding sites and you get into the cell and do it?
Sure. So PXR, rifampicin activates PXR, as he said, which is also a nuclear receptor. So this would be, instead of targeting the binding site of the androgen receptor, the PXR would suppress it, not through the binding site pretty much. So the treatment kind of targets one thing, it has an effect on another instead of targeting the binding site of the androgen receptor directly. So. Anyone else?
Okay, thank you, Brock.